Welcome to the BioInsights, where science comes alive.
Okay, today we are going to explore a hidden world that is literally right under our noses. From that incredibly stubborn layer of morning plaque on your teeth, all the way to the gross, clinging slime you find in your kitchen drain, we're dealing with something far more complex than just a random smudge of dirt. What we are actually looking at are highly organized, unseen, microscopic cities. So let's just dive right into this hidden world and demystify exactly what these structures are and how they practically govern our environment. It's a great question, isn't it? Like, why does it take so much intense elbow grease to scrape that slime away? Well, when you're aggressively brushing your teeth or scrubbing down a kitchen drain, you aren't just wiping away loose dirt or some isolated germs. You are actually engaged in a full-on battle to dismantle a deeply entrenched, highly fortified biological fortress. Section 1. The Microscopic City. All right, to really understand this fortress, we first need to completely change how we picture bacteria. For centuries, scientists just pictured them as these solitary, free-floating entities wandering blindly through the world. But actually, living as a single drifting cell, a nomadic bacterium, is incredibly dangerous. It leaves them totally vulnerable to UV radiation, starvation, predatory immune cells, and, of course, modern antibiotics. So to survive, the vast majority of bacteria in nature completely abandon that solo nomadic lifestyle. Instead, they seek out surfaces and cooperate to build a biofilm community. By teaming up, these simple, single-celled organisms transform into a collective, heavily defended biological city that can easily survive environments that would instantly wipe them out individually. And just how effective is this collective defense? Get this, bacteria tucked safely inside a biofilm are typically 200 times harder to kill with standard antibiotics or disinfectants than their free-floating counterparts. 200 times. As long as they remain alive inside these shelters, they are a seriously enduring threat. Moving on to Section 2, Building the Slime Fortress. So how exactly does a tiny, completely vulnerable lone cell physically build such an impenetrable shield? Well, it all starts on a moist surface that's coated with a thin layer of proteins and sugars. Curious, free-swimming bacteria propel themselves toward it using their tail-like flagella, just bumping into it to make a weak, reversible attachment. But if the conditions are right, something absolutely amazing happens. A genetic switch actually flips. The bacteria deactivate their swimming genes, and they switch on their biological machinery for building. They go from a vulnerable nomad to a fully committed architect. These newly anchored cells rapidly divide, forming microcolonies, and begin excreting the sticky biological mortar known as EPS. Eventually, a mature fortress will even release planktonic scouts back out into the fluid to go off and colonize brand new areas. The whole cycle just repeats. Now, here's where it gets kind of wild. The physical makeup of this mature fortress is completely counterintuitive. The actual bacterial cells, they only make up about 5 to 15% of the total volume. The vast majority of the community, an overwhelming 85 to 95% of it, is just that sticky mortar, the extracellular polymeric substances, or EPS matrix. And listen, this matrix is definitely not just random unstructured slime. It is a highly engineered multi-material structure. You've got long sugar chains acting like a rigid structural scaffold. Then dense lipid molecules come in to form incredibly effective waterproof barriers. And amazingly, some bacteria will literally release their own extracellular DNA into the environment to weave through the matrix like molecular rebar, which vastly increases the physical strength of the entire colony. It is specifically designed to withstand intense mechanical and chemical stress. Section 3. How bacteria talk. As you can probably guess, running a massive construction site takes some serious coordination. To organize all this, the colony relies on an invisible chemical communication network called Quorum sensing. Instead of acting individually, the bacteria release these small signal molecules. As the population grows, the signals accumulate. Once they hit a specific threshold, boom, it triggers the microbes to synchronize their behavior and operate as a single, unified organism. And this isn't just some one-size-fits-all language either. Gram-negative bacteria use AHL molecules, gram-positive use peptide signals, and there's even a universal auto-inducer system for cross-species communication. It's like a microscopic UN translator. This brilliant communication leads to some highly synchronized survival tactics. The coordinated cells literally build internal water channels to route nutrients in and wash waste out.
They establish these distinct zones deep down in the center where oxygen levels drop, which allows some microbes to become dormant persister cells. These are cells that can easily survive drugs that are designed to only target actively dividing bacteria. Meanwhile, nearby active cells use their incredibly close physical proximity to strategically swap antibiotic-resistant genes, like trading cards. Through all this communication, they create an incredibly intricate biological shield.
Section 4, the threat to us. So we've seen how incredible their architecture is, but what are the real-world consequences when these microscopic metropolises actually invade our lives? Well, we can categorize the threat by looking at where these biofilms thrive. In medical environments, they can grow on living tissues, like chronic wounds, or on non-living, abiotic materials. Think pacemakers and plastic catheters. Here, they block immune cells and cause intractable infections. On the flip side, in industrial and aquatic settings, like your drain lines and city water systems, they narrow pipes, which causes sluggish flow, extremely costly corrosion, and, of course, those foul odors.
In a medical context, the threat is frankly severe. When a biofilm sets up shop in a wound, it turns into, exactly as this quote describes, a slimy, gooey castle. The dense EPS matrix physically slows down incoming antibiotics, and it chemically traps them before they can ever reach the deeper cells. And worse, it completely blocks your body's natural cleanup crew, the immune cells. This resilient colonization cycle makes medical implants, like artificial heart valves, incredibly vulnerable. Once they are established on a metallic surface, the infection is often downright impossible to eradicate without complete surgical removal. So the absolute crucial point here is prevention. Simple, thorough, early wound irrigation is critical to wash away those sneaky invaders before they can anchor down and build their castle.
But let's bring it closer to home and see how this science translates into actionable plumbing defense. In your pipes, as that 3D biofilm layer thickens up, it literally narrows the pipe's diameter and starts secreting bacterial byproducts that smell awful. But you can definitely fight back. Routinely flush your drains with hot water. Use enzymatic cleaners that are specifically designed to break down that sticky EPS matrix glue. Whatever you do, avoid feeding the microbes by keeping grease and food scraps completely out of the drain. If you happen to be building or renovating, choose pipe materials that are much less friendly to microbes, like copper. And hey, since they absolutely thrive on the extra nutrients found in hard water, installing a water softener can also literally starve them out. Routine maintenance stops the slow drains way before the fortress gets too big to handle.
Section 5. Harnessing the good slime. Now, before you think it's all doom, gloom, and clogged pipes, there is a totally different side to this. Human engineering has actually allied with these bacterial cities for environmental good. We figured out that this exact same cooperative power is a highly valuable tool for large-scale engineering and food science. In massive wastewater treatment plants, engineers intentionally cultivate industrial-sized biofilms right in the settling tanks to aggressively digest organic pollutants. Out in the ocean, bioforming bacteria like Alcanovorix borchomensis actually eat harmful chemicals, purifying the water to protect marine life. In agriculture, beneficial biofilms wrap around plant roots to shield crops, like pea plants, from disease-causing microbes like the Fusarium fungus. And of course, let's not forget food production. We literally use biofilms to cultivate kombucha teas and generate those amazing sweet and sour flavors we love. Because biofilms are so deeply integrated into every aspect of our world, modern research is accelerating at a breakneck pace right now. Scientists are aggressively targeting quorum sensing to try and prevent infections before they even start. We are seeing amazing breakthroughs where artificial intelligence is being used to model these highly complex bacterial behaviors, and cutting-edge nanotechnology is literally being deployed to physically block their chemical communication signals entirely. Decoding this architecture is exactly how we are going to defeat intractable diseases and scale up sustainable green industries for the future. Which leaves us with this final kind of mind-blowing thought. We live on a planet that is fundamentally governed by these cooperative microbial ecosystems. From the pipes hiding in our walls to the pacemakers in our hearts to the very food we consume, are we truly ready to fully master the architecture of the microscopic metropolis? It is an incredible invisible frontier, and honestly, we are just scratching the surface. Keep questioning, keep learning, and thank you so much for joining me.